Hello everyone, my name is Rahwa Tomas and this is my PowerPoint presentation on H. Salminicola for the Bio 282 lab section on Tuesdays at 10:30 in the morning. The news article I used for this assignment is from CNN World and the primary literature article is from PNAS. To start off, what is H. salminicola? Its full name is Haenaguia salminicola is a myxozoa which is a type of parasite that is part of the cnidarian family and shares relations with seawater organisms such as jellyfish and coral. H. salminicola lives inside their host salmon. They also live in other freshwater organisms such as trout. They are also most known for not needing oxygen to live. Instead, they quote-unquote steal nutrients that were already made rather than consume oxygen directly. These parasites live in low oxygen environments where they are not able to breathe properly. In order for them to survive, they must be in a vacant area of their host which doesn't contain air and allows them to quote-unquote breathe and survive. Researchers claim that H. salminicola isn't the last oxygen-free parasite, and there are many more with unusual modes of existence. Here is my attempt at drawing what the parasite looks like. And next we can take a look at taxonomy. At first, this taxonomy may seem overwhelming at first glance, but it's self-explanatory for most parasites. Since we are learning about H. salminicola at the beginning of the presentation, we already know it's a myxozoa and it's part of the cnidarian family. If you took some time to locate it, the family of Cnidaria is in the light pink area and below is myxozoa, in which H. salminicola is located. Researchers based their phylogenetic analyses from a source of 78 nuclear ribosomal protein encoding genes. They also compiled organisms that were verified by the taxa representative of eukaryotic diversity. Myxozoans have impressive simplifications during adaptation, both morphological and genomic, from the cnidarian ancestor. It's interesting how most of the findings are not ancestral but could be caused from secondary losses. However, due to the increasing rapid evolutionary rates of myxozoans, it has led them to the reduced ability to find typical eukaryotic genes. And this leads up to the structure of H. salminicola. As we delve into their structure, it has been noted that it has great resemblance to mitochondria in terms of DNA. Vital enzymes that are necessary for respiration, H. salminicola was not capable of producing them. Not to mention, researchers couldn't even find a trace of mitochondria while observing its genome, which was surprising at first. However, researchers were able to identify a cristae structure in which it is able to imitate the inner folds of mitochondria but doesn't function the same way as mitochondria. Looking at the image on the left of the microscopic display of H. salminicola, the cristae researchers classified are huge white spaces circled in red. And next, after seeing H. salminicola structure, it's important to understand how it functions. For H. salminicola, they are unable to perform aerobic cellular respiration. Seeing the three microscopic images taken, it's clear to see that there is no mitochondria. Many researchers claim that since salmon muscles are accustomed to living in the low oxygen environment and H. salminicola lost its ability to generate oxygen, indicating that these eukaryotic features aren't familiar among organisms. With the loss of the mitochondrial genome, some of the mitochondria have formed to become mitochondria-related organelles, or also known as MROs. Not only did they lose their mitochondrial genome, but nearly all their nuclear genes which are required to go through transcription and replication. And last but not least, I want to discuss H. salminicola versus other parasites. Researchers figured out that mitochondrial DNA polymerase subunit gamma-1 gene is a pseudogene, which is a section of a chromosome that is an imperfect copy of a functional gene. It contains 3-point mutations that create premature stop codons, which led to the principal conclusion that H. salminicola lost both its mitochondrial genome and mitochondrial DNA replication machinery. Once the mitochondrial genome is lost, it would have a significant impact on aerobic respiration since animal mitochondrial genomes code proteins that are crucial for the electron transport chain. To confirm whether the loss of the mitochondrial genome is indicated as a loss of aerobic respiration, in H. salminicola, researchers particularly focused on all genes in complex 1, 3, and 4, which are classified on most myxozoans, but not H. salminicola. In conclusion, the news story accurately portrayed the findings of the primary literature article, but only has general information and has less detail about H. salminicola, unlike the primary literature article, which is more complex and is very detailed. Thank you for taking the time to listen to my presentation. Here are my references of the news article and primary literature article, and of images and other sorts. Thank you.